Here is our live for today. So the details of the presentation could be found in this LS paper, and the research is supported by the NIH grant. This project is joint worked with Aaron, who is a postdoc at Columbia University, Michelle, who is a graduate student at UMass, and Patrick, who is my advisor, assistant professor at UMass. For motivation, this graph may better summarize our motivation. It shows a tumor which composed of 3 clone subpopulations. 3 samples are obtained from the solid tumor, 2 are bulk, sample 1 and 2, 1 and 3, and 1 is a single cell, sample 2. So these 2 subpopulation genotypes are unknown. So we want to jointly infer bulk and the single cell sequencing data, and thus infer the subpopulation type genotypes and the posterior distribution of subpopulations from related individuals. The reason we want to include multiple individuals is that we think it could be beneficial for clinical trial or other researches that need selected group. So we developed 2 methods for that. One is HDP, the hierarchical de-Richter process model, which captures the whole picture and have flexibility to the different hyperparameters, but it needs expensive computation. The other one is HGP, the hierarchical gamma Poisson model. It can be sample fast and lose some flexibility. For the problem setting up, we assume we sample some multiple individuals from the population and sample multiple biopsies from each individual. So for the typicity, we assume the biallelic, or we only assume biallelic variant with the major allele big A and minor allele small a. So that we have 3 genotypes, 2 big A, 1 big A, 1 small a, and 2 small a's. So the only observed data will be the read as IJLR, which means the R's read in the ith individual, J's biopsy, and L loss curves. Then we have a genotype-based matrix T. So the epsilon here is the error rate, which can be tuned to reflect the different error rates for bulk and the single cells. So for example, so if the true genotype is 2 big A, then it is not chance the read is A, but there also is some small chance it could be a small a. So here is the complete model HDP. The HLK here is the genotype, so which comes from a multinomial distribution, and we use the truncated Dirichlet process as hierarchy, and find the final read as IJR, then it's drawn from a categorical distribution. Here is a graph representation of the model, and we implement a gift sampler, which requires some maturity testing steps. HGP is a little different. We count the read of the data, and thus the observed data is count of the read as death locus. So it requires the same transition matrix for all samples, but it can handle larger scale of data set. So here is the complete HGP model. And as we can see, we use gamma distribution instead of the Dirichlet distribution as hierarchy, and observe the data from a Poisson distribution at last. So here is the graphical representation of HGP, so which is similar to HDP, but it has less hyperparameters to control. So here is the sample algorithms we use, which use the augment and marginalized procedure. So you can derive full analytical form of posterior distribution, and thus can be fast and handle large data set. Then here are some results for both simulation and real data analysis. So for simulation, data were simulated via a parametric model using the Dirichlet distribution. So we filter to a small data set of a mixture of bulk and single cell samples. So here we use like 6 individuals and 4 biopsies per individual, 1 bulk, 3 single cells, and HDP find all true genotypes with a small KL divergence at the different levels. And the posterior distribution shows the true value, and the posterior distribution estimates from the simulation model. So this result indicates that the model is able to identify the true subpopulation genotypes, and the posterior distribution are appropriately uncertain relatively to the amount of data. We also compare our model to other state-of-the-art methods and the apps, which also show our model can better performance on this kind of data for clustering and estimation. So we also did some sensitive analysis to show that our model can handle different situations. And for HGP, it can be used for largest data sets. So we fit to a stage individual and localized single cell samples per individual. And also the HGP identified all true genotypes with a small KL divergence. Then for the real data analysis, we use the acute lymphoblastic leukemia data, which have 6 patients and a total of 79 single cells and bulk samples. So the HDP identified 3 major components for patient 6 and show large probability on the components that only have plaque mutation for single cell S101, so which is similar to the original data set. We also apply HGP to larger data set, which has more loci, and it also can identify different components. Use HGP, we can also construct a co-occurrence matrix to estimate which genes mutation will happen together. So the graph shows the results for patient 1. One of the high connect gene, TTN, here has been recently reported as the most frequently mutated gene in a pan cancer cohort, and it's associated with increased tumor mutation burden. So but while these co-occurrence inferences are subjective, it may lead more validation experiments. For the future work, we will apply the 2 models to transperson sequencing data to identify the conditional essential genes networks. So that's all. Thank you.